It's a great pleasure and in a way also a great opportunity to present AFRIBIOTA to the Pasteur network. I will try to be very brief because I wish to leave most of the capacity of presenting to Dr. Violeta Moya Alvarez and Dr. Henko. So I would just like to give you some sort of a retrospective and perspective of AFRIBIOTA. The retrospective aspect is, in a way, the history of AFRIBIOTA. It emerged, on my side at least, to start with the original concept by the frustration of working on bacterial pathogen, but realizing that there is not just pathogens in life, and there are also commensal microorganisms, which are very important to consider. And at the time, which is about 10, 15 years ago, it became obvious that this community of microorganisms, especially the one that inhabitates the intestinal tract, the small intestine, also, of course, the large intestine, might be altered by certain environmental conditions. And this alteration may lead to what is usually called dysbiosis. And in this sense, we are joining the concept of pathogens, but now instead of considering one single pathogen like Shigella, which is destroying the intestinal epithelium and causing pain and bloody diarrhea, and also consequences on nutrition, because it's mostly prevalent in young children in their first years of life. There was also the concept of dysbiosis, which was itself able to cause diseases, but diseases that are not like acute disease, like an infection, but more sort of slow processes of degradation. And then came the idea that changes in microbial population in young children in the most impoverished areas, and I had been sensitized to this by my activities at the WHO, for instance, for the development of vaccines against enteric infections. This may have an impact on child nutrition and would be probably an important parameter, if not a cause, of what's usually called stunting. Stunting is a developmental issue in children after birth. It causes growth retardation, but not just, growth retardation, which is an issue, but they can catch up in a way in the future. But it also causes hypersusceptibility to microbial infections, and the death rate of these kids is multiplied by a factor of 4. And also neurodevelopmental disorders, not very severe, but something that often affects acquisition of knowledge. I mean, in school, these kids have lower capacities than the properly nourished ones. So stunting, we thought, was a burden for the society in the countries that are affected. I mean, in intertropical Africa, it's considered in general that the incidence of stunting is about 40%, which is huge. And we thought that there was an opportunity for us, the Institut Pasteur in Paris, with the experience we had on basic mechanisms of subversion of the intestinal mucosal tissues, and colleagues of the Pasteur network. It wasn't called Pasteur Network at that time because we started about 10 years ago. This was yet the Institut Pasteur du Réseau, but we thought it was an opportunity to build something together. And the program was actually a sort of triangular collaboration with the Institut Pasteur in Bangui, with Dr. Emmanuel Nakune, and the Institut Pasteur of Madagascar, with Dr. Rin Ramandranara. There are long names in Madagascar. And all these people were extremely impactful in the development of AFRIBIOTA. It was really both at the medical, scientific, and I would say ethical level an experience of how one can manage at equal value and contribution collaboratory programs together. So the idea was it was a sort of sectional study at that time on children between 3 and 5 years of age, detecting stunted kids in comparison with controlled kids. And then Violeta will get into the detail, try to figure out what kind of and what level of dysbiosis these kids were suffering, and whether this would open possibilities for intervention, either prevention or treatment in the future. So that's what AFRIBIOTA was. This triangular adventure. We also had partners, including in Canada, the group of Brett Finlay at University of British Columbia, Vancouver. We had neurodevelopmental people in the process. So it was kind of a multidisciplinary study. And the person who initially set all this together and was in charge of the coordination was Dr. Pascal Vonesh, who was a postdoctoral fellow in my lab. Very interested also in health issues in the low-income countries. And Violeta, who a little later joined us and not only collaborated and participated in AFRIBIOTA, but also developed her own program, Mythica, which was based on study of diets of mother children after birth and looking in a way what could be the participation of mother's malnutrition in this development of stunting and also whether stunting was starting very early in life and not something that was coming later, like at the age where it had been studied initially in the AFRIBIOTA program. So this is a coordinated program, and we thought that it might be a nice opportunity to strengthen links between the different members of the Pasteur network, at least those in countries that are, affected by stunting. So to us, it's an open program. We are hoping that other colleagues from other institutes will help. The more we will be, the better we'll be able to accumulate funding. We are actively, it's a lot of our activities to try to find funding to support not only the continuation, but the development of AFRIBIOTA. And so it's a hope that all together we'll be stronger and will gain even more credibility among national or international institutions to be able to support the continuation of what I consider as a fantastic human, medical, and scientific adventure on its development over the last years. So thank you very much for attending, and I'm happy to leave the floor to Dr. Violeta Moya Alvarez, who will tell you, again, history, present, and future of AFRIBIOTA and its associated activities. Thank you very much for your attention.
Thank you very much, both of you, for this presentation. So when we were asked about presenting the AFRIBIOTA program, it was quite challenging because it's been a long story. We've already conducted 3 different studies and we want to conduct another one. So I'm going to give you the plot of this adventure, but also I would like you to show all the efforts of all the teams, the different teams on the field. So there are some scientific results, but please, if you have more scientific questions, don't hesitate to leave it for the end and we will be glad to answer them. So first of all, it doesn't work. So first of all, I'm going to present you and to introduce the concept of environmental and enteric dysfunction, because AFRIBIOTA, the entire AFRIBIOTA consortium and programs are based on preventing treating and understanding this disorder, this disease. Then I will give you some results about the different AFRIBIOTA studies that we have conducted so far. And then Dr. Nkokinding will give you some other inputs on how AFRIBIOTA 2 is ongoing at the moment on the field in the Central African Republic. So first of all, what is EED, or environmental enteric dysfunction? So it's an acquired chronic inflammatory syndrome of the small intestine. And the history of this disease stems actually from colonial times and also from war, because it was first described by English physicians in India that were keeping the Italian prisoners of war during World War II. And Italians that were kept prisoners in India started not only to emaciate, but also to have severe but low-grade intestinal inflammation. And that's spontaneously resolved when they went back to Italy. So that was, I mean, the first time that, let's say, so Western physicians described this condition. And later on, it became popular again in the 50s, 60s, when Kennedy deployed the Peace Corps. And actually, Americans going to the Southern Hemisphere mainly had the same symptoms, as these Italian prisoners of war. And for me, it's very controversial and shocking to see that there is a disorder and a disease that was, and that is still unlikely, very prevalent among the Southern populations, but it was first described in Western populations, which tells us a lot about history, and we should learn from that. So about what is known about the physiopathology of EED? So first of all, we know that in response to a highly microbiology polluted environment, poor nutrition, and frequent infections, dysbiosis can easily emerge. So gut dysbiosis is characterized either or several factors that are pathogen expansion, reduced diversity of this microbiota, or a loss of beneficial microbes. So once a gut dysbiosis is established, this might pave the way to the emergence of environmental enteric dysfunctions, which is characterized by immune infiltrates, but also intestinal inflammation, increased permeability, increased microbial translocation, and nutrient malabsorption. In turn, this results into EED symptoms, which are mainly growth faltering, oral vaccine failure, cognitive impairment, and anemia and nutritional deficiencies. From the public health perspective, this represents a huge burden of disease. We know that there are more than 150 million children that are stunted. So just for the definition, stunting is defined as a length for age shorter than 2 standard deviations of the WHO growth curves. And we know that stunting is associated with over 370,000 deaths in under 5s alone globally. Also, EED is correlated with oral vaccine failure, as I said. Among EED infants, rotavirus vaccine fails in almost 70% of the infants, and around 20% of the infants with EED, that's the failure associated with polio vaccines. In addition, as previously Philippe said, we have an impaired neurocognitive development with an average 11 IQ points less by age 8. And here we have some histological patterns that we can see from the typical brain cells and impaired brain cells with EED from some autopsies that we got. And finally, it's also malabsorption syndrome. And we know that globally at present, there are 1 in 2 children that are that have some kind of micronutrient deficiency and in 3 women. And what does all this tell us? That tells us that EED is not only an inflammatory syndrome, but as Philippe already said, a developmental challenge, because it will entail severe long-term repercussions for infant health. So, as I told you, in the AFRIBIOTA consortium, we aimed to first understand and describe what was ongoing, like the physiopathology of EED, that was AFRIBIOTA 1. Then we conducted a preventive intervention that I'm going to go more in depth in AFRIBIOTA 2. And then I will only say some words about AFRIBIOTA 3. So first of all, that's Pascal Voulache and Philippe Sansonetti, both the co-PIs of the first AFRIBIOTA study. And the main objective of the AFRIBIOTA study was, as I said, to shed light on the interactions between dysbiosis and stunting, or EED, in children between the age of 2 and 5 years in the Central African Republic and Madagascar. Why between 2 and 5 years? Because it's when stunting is first diagnosed in low and middle income countries. It was a case control study where they analyzed 100 severely and moderately stunted children, and they compared these children to 260 children per country. And they mainly did 2 great discoveries. First of all, EED was highly associated with the presence of enteropathogens in the duodenum. And also there was small intestinal bacterial overgrowth with oropharyngeal species in the duodenum, which was kind of a surprise. And second, there was this overrepresentation of oropharyngeal species as well in feces. And of course, there was also an overrepresentation of enteropathogens and pathogens. And more specifically, in the clinical study, EED markers were associated with elevated fecal levels of Fusobacterium, Megasphaera, and Collinsella, and lower levels of really strict anaerobic bacterial producers. And then taking these isolates from the clinical study, they performed in vitro and animal models. And actually what they found was that these bacterial isolates were associated with the reduced abundance of classic small intestinal bacteria, but also with decreased lipid absorption and increased cell layer permeability, directly associating then these bacterial isolates from oral origin with stunting. So that was the publication, the main publication of the study in PNAS. And so first we can say, okay, if the problem is oral bacteria, let's do something about oral hygiene to prevent this aberrant colonization of the gut. But before we go into AFRIBIOTA, that was the idea of AFRIBIOTA 2, but let me just present you some other evidence from another study, which is the MITIGA study that we also performed in the Central African Republic. So the idea of MITIGA, which was the mother-infant transfer of microbiota in Central Africa, was really to go upstream. Because, as I told you, AFRIBIOTA 1 was conducted among infants from 2 to 5 years. And actually, what I was really looking forward was to see what happened before, because we noticed in AFRIBIOTA 1 that 2 years was definitely too late. Therefore, we conducted this longitudinal observational cohort of 50 mother-infant dyads in Bangui with a BTR funding, which was of like Present Spark, but before it was called PTR. And we collected blood samples at delivery from the mother and the infants, and also maternal stool and vaginal swabs at delivery, and then oral swabs and infant oral stool at delivery 1 week, 1 month, 11, 18, and 25 weeks. We collected also blood at 3 and 6 months. so at 11 and 25 weeks. And we also collected breast milk from the mother at 1 week, 1 month, 11, 18, and 25 weeks, in addition to classic epidemiological data, including also antibiotic intake, diet assessment, and we also analyzed blood test, sorry. So, and that's the infant characteristics from birth until 6 months. And what I want you to see first is that after food diversification is the moment in which the infant starts to receive other food other than human milk. And it usually takes place, at least in Bangui, between around 2 months, 2 to 3 months. And actually, we see here the stunting rate, I don't see my mouse here, get from 70% to 23%, and then increase again to 35%. And so here we don't see it so markedly because we have like the systematic visits. And of course, not all infants had food diversification at the same time, but when you see it, you see a clear association between food diversification and the increase of stunting. Also, another important result was anemia, because you see that after, again, food diversification, anemia increases, but mainly all micronutrient deficiencies, meaning iron deficiency that goes from 3% to 21%, and also vitamin A and vitamin C, which then start to increase already at birth. And we have 1/3 of the infants who were vitamin C deficient at 6 months, and over 70% were vitamin A deficiency at 6 months. So in conclusion, we see that there's really an important nutritional burden already at 6 months when the infant just start to live. So when we analyze the microbiota of the infants, the oral microbiota, because here I will only present everything which is regarding to oral microbiota, because that's the thematic of AFRIBIOTA 2. We saw that there was borderline difference in the oral microbiota between children with ongoing inflammation defined by CRP and non-ongoing inflammation. So that this inflammatory syndrome of EED is, we could say that it's already reflected also in the oral sphere. Second, we saw as well that there was a difference of between the microbiota of stunted children at 6 months and the microbiota of non-stunted children at 6 months. And again, that's after food diversification. And finally, and that's a crucial point, there was also borderline significant difference in the oral microbiota during the entire follow-up, depending on the human milk type of the mother. What does this mean? So, depending on the secretor and Lewis type, which are antigens that are coded by the FIT2 and FIT3 genes, we have 4 different human milk types. So, secretor positive and Lewis positive are milk type 1, secretor negative, Lewis positive is milk type 2, secretor positive, Lewis negative is milk type 3, and secretor negative, Lewis negative is milk type 4. So we saw that infants of human milk type 3 mothers were less likely to be stunted compared to those of human milk type 1. And also something that is coded by these genes FIT2 and FIT3 are human milk oligosaccharides, meaning that depending on your secretor and Lewis status coded by these genes FIT2 and FIT3, you will have different patterns of human milk oligosaccharides in human milk. And HMOs are, this human milk oligosaccharide feed we will call HMOs, are complex, non-digestible glycans that are naturally abundant in human breast milk. And why are they important? First of all, because they are very powerful prebiotics. Second, they have antibiofilm properties. Third, they increase the barrier function. They also associated to reduce inflammation. They also influence gene expression, and they are also very meaningful for brain and cognitive development. And SL6, sorry, and SL3, for instance, are also associated with lower incidence of enterocolitis, necrotizing enterocolitis among preterms. And actually, at present, there's a phase 3 trial with these HMOs ongoing to fight and to prevent NEC in the United States. And that's our idea, maybe, is to use these HMOs in AFRIBIOTA 3 to prevent the aberrant colonization on EED. Because indeed, this what we found in the Mythica study, what there was, these HMOs were associated to first reduce risk of stunting, better length for age, better growth, reduce pathogen, the relative abundance of pathogens such as Klebsiella or E. coli, and increase relative abundance of bifidobacteria such as Bifidobacterium longum or Bifidobacterium breve. Here, it's you can see the multi-level mixed model that we performed on stunting risk from birth until 6 months. And as you see here, the risk of stunting, well, here's one, was 100 times less in the milk type 3 infants compared to milk type 1. And here we have certain HMOs that were also correlated, this one with increased risk, but we have also LNNT, which was a protective HMO. And of course, there were adjusted on hygiene and the human, the age of the infant, sorry. So in conclusion, from the AFRIBIOTA 1 and the Mythica studies, we have 2 main points. First, we need to consider oral microbiota in the study and the prevention of EED. And second, there are other determinants of the oral microbiota that we need to consider, such as antibiotic consumption, inflammation, as I show you, that the oral microbiota differed also depending on inflammation, but also human milk. Therefore, when we were writing the AFRIBIOTA 2 study, we first said, okay, we know that stunting is associated with oral dysbiosis. So first of all, what we have to do is to characterize this oral dysbiosis, because until now we had mainly focused on the gut, and we didn't know what was going on the oral sphere, and therefore we wanted to characterize first of all this oral dysbiosis. Sorry, and then we wanted to analyze also the dynamics of this oral dysbiosis and how these evolved during the first year of life, and especially during the food diversification periods. And therefore, our second research question was, which are the etiological pathways leading to oral dysbiosis? And we wanted to characterize these origins and dynamic of this oral dysbiosis. And finally, of course, we wanted also to tackle this oral dysbiosis associated with EED, and therefore we conceived this hygiene-focused oral intervention, and coupled with a nutritional intervention, and this Dr. Ingo will tell you about later. So for all these reasons, the research objective of AFRIBIOTA 2 are first to demonstrate with a specifically designed trial in Bangui that nasopharyngeal and oral hygiene education might prevent stunting. Second, to investigate the oral cavity as a microbiological hub that monitors not only oral microbiome assembly, but also its dynamic, considering as well the other determinants that I just named, namely the maternal and child hygiene, nutrition, and breastfeeding. And finally, because to nucleate the conditions for the development of a global program of education to oral health in the Central African Republic, because it's true that oral health is kind of a neglected health dimension in Central Africa so far, and also in West Africa and many other areas. So therefore, we wanted to conduct this clinical trial, this intervention trial, and we will collect the samples of the highest and lowest length for age and the highest and lowest growth patterns of this oral and also fecal microbiota. And what we'll do with them is we will perform the metagenomic analysis to characterize during the first year of life all these samples, but not only among infants, but also among these families. And then once we will have this metagenomic analysis, we will focus on the most abiotic and dysbiotic microbiota and discordant families, namely, for example, families where the parents clearly have dysbiotic microbiota and the infants an abiotic microbiota, to analyze the transmission patterns between them, or what are the factors that have prevented the infant from getting sick. So for our first research question on oral dysbiosis, to characterize this oral dysbiosis through the first year of life, we are using two different techniques, as I told you. First of all, culturomics analysis that are performed by Dr. Ina and Dr. Jean-Marc Collard. Dr. Ina is in Bangui. She's the doctor that is leading the bacteriology part of the AFRIBIOTA study, AFRIBIOTA 2 study at the Institut Pasteur de Bangui. Jean-Marc Collard is in Paris, which is the PI who is the PI for this work package. And now Ina is coming to analyze and do the multi-TOF analysis at Dr. Jean-Marc Collard's lab at the Institut Pasteur. And then Pascal Vounache will perform the SHOT1 analysis that will characterize this oral dysbiosis. Sorry. Our second research question about the ethiopathological pathways leading to this oral dysbiosis, mainly during food diversification, will be analyzed with meta-IC, which is a very powerful and cutting-edge tool to perform microbiota analysis, and this will be performed at Romain Cosul's lab at the Institut Pasteur in Paris, together with Marcel Marvouti. And finally, the clinical part and the interventional trial is performed in Bangui by Dr. Yannick Kenko and Dr. Sheila Yangere, that he will present just afterwards. But there are also other people, of course, that are very fun, that are working with us, and they're very, very helpful, namely Cassandre from the, CICR, the Translational Research Center at the Institut Pasteur, but also Alexandra Vautreau-Wunter at Institut Pasteur de Bangui. So that is the entire consortium. We got PTR for this study, and also INF. And we are from different labs. We employ different complementary techniques. And of course, the PIs are also Philippe Sansonetti and Emmanuel Nakune. Before Dr. Enko presents you the clinical and the epidemiological part, I just wanted to give you a small focus on the disease burden of the Central African Republic. In case you don't know it, the Central African Republic is a very big country, but with a small population, I would say. It's about 6 million people now, but a very high fertility rate, and the population is expected to double by 2050. Life expectancy is about 22 years old, and the large majority of people die, 66%, from communicable maternal, perinatal, and nutritional conditions. 23% of the causes of death are non-communicable diseases, and 10% injuries. It is estimated that 40% of children younger than 5 years are stunted in the Central African Republic. And just to give you also some data about anemia among women of reproductive age, the prevalence is also very high, almost 50%. So now I hand it off to Dr. Nku so that you present the trial. Are you there? Dr. Nku?
Oui. Merci beaucoup. Pour atteindre en fait les objectifs fixés par cette étude, nous avons opté pour une étude interventionnelle longitudinale randomisée avec 2 groupes. Un groupe intervention qui reçoit une intervention d'hygiène orale et nasopharyngeale associée à une supplémentation nutritionnelle à base de suppléments nutritionnels lipides de petite quantité. Et également, on a un groupe de contrôle qui reçoit uniquement la supplémentation nutritionnelle. Il est important de noter que le critère de jugement principal est le score longueur pour âge à 12 mois. Le critère secondaire, c'est la prévalence du retard de croissance. Pour en fait dimensionner notre étude, nous sommes appuyés sur les résultats d'une étude de Médecins sans frontières au Niger en 2009, réalisée par Isanaka et collaborateurs, qui avait démontré en fait une amélioration moyenne de la longueur pour âge de 0,14 à 12 mois grâce à la supplémentation nutritionnelle. Nous avons donc fait l'hypothèse qu'une intervention d'hygiène bucco-nasopharyngée pouvait apporter un bénéfice supplémentaire de 0,2 points de la longueur pour âge. Avec donc une puissance de 80% et un risque alpha de 5% bilatéral, le calcul a conduit quasiment à un effectif de 255 nourrissons par groupe, soit un effectif total de 450 couples mère-enfant. Notons que le suivi en fait des participants sur le plan pratique prend en compte une équipe en fait qui est constituée des relais communautaires à la base, beaucoup plus sur le terrain, qui sont en première ligne quasiment. Et secondairement, on a les médecins investigateurs, moi-même et docteur Sheila Yangere, et une équipe de laboratoire aussi qui quasiment tous travaillent en symbiose. D'abord, pour les relais communautaires, ils sont responsables en première ligne du recrutement des mamans, en fait, en vérifiant que les critères d'inclusion sont bien respectés. Une fois que ces conditions sont réunies, les médecins investigateurs descendent sur le terrain pour contrôler en fait qu'effectivement la maman remplit les critères d'inclusion. Et si ces critères d'inclusion sont remplis, on procède maintenant à son inclusion effective. Et donc, pour le moment, nous sommes à 446 couples mère-enfant avec 225 en intervention d'hygiène et supplémentation nutritionnelle et 221 couples de contrôle. Le suivi comprend entre autres les mesures anthropométriques dont nous procédons à la prise des mesures anthropométriques, entre autres le poids, la taille. On prend également le périmètre crânien. On procède également au remplissage de certains questionnaires, des questionnaires qui sont beaucoup plus en lien avec les mesures d'hygiène parce que c'est vraiment un point phare pour nos groupes d'intervention. On procède également au remplissage des formulaires de rappel de 24 heures et les formulaires de fréquence d'alimentation. Il est également question de l'examen du bébé. Comme il s'agit d'une étude qui prend en compte l'hygiène buccale, on examine la bouche de l'enfant. On procède aussi, au-delà de ça, au prélèvement des écouvillons orales de l'enfant, on prélève les selles de l'enfant, et également les écouvillons oraux des membres de la famille. Au-delà de ça, il y a également les prises de sang qui se font au niveau de l'Institut Pasteur. On le fait à la naissance pour les mamans, on le fait à 6 mois pour le bébé et également à son 12e mois de vie. Il est également important de noter qu'au-delà de ces 7 suivis normaux, il y a également 2 suivis supplémentaires qu'on réalise en plus lors de la diversification alimentaire. Donc, lorsque la maman introduit d'autres aliments que le lait, on procède également à une visite de suivi et une 9e visite de suivi après le sévrage, une fois que la maman a arrêté l'allaitement au sein. Pour ce qui concerne les groupes d'intervention, notre focus est beaucoup plus mis sur les groupes d'intervention en éduquant aussi les mamans, parce que c'est aussi un aspect qui est très important, sur comment suivre le développement de la bouche de l'enfant. On explique aux mamans la formation de l'apparition normale des dents, comment prévenir les infections, comment reconnaître les signes d'infection. Et pour cela, une fois qu'il y a éventuellement une infection au niveau buccal, par exemple une candidose buccale, le projet se charge de prendre en charge ces enfants sur le plan thérapeutique. Il y a également les actions qui sont menées au niveau de la bouche qu'on est censé expliquer aux mamans, comment nettoyer la bouche après chaque repas, laver la bouche après chaque repas ou allaitement. C'est important à expliquer comment bouillir les repas, l'intérêt d'éplucher les repas, l'intérêt de libérer les ongles de l'enfant, l'intérêt de nettoyer le nez de l'enfant au sérum physiologique au moins 1 fois par jour, l'intérêt de nettoyer également les sucettes au moins 2 fois par jour, très régulièrement. On met également un focus sur tout ce qui est conseil en lien avec les habitudes de l'enfant. Il est important d'expliquer aux mamans quel est l'intérêt de réduire en fait l'usage des sucettes et du pouce, l'intérêt de réduire en fait les contacts de l'enfant avec les animaux. Et également, on explique aux mamans tout ce qui est en lien avec la nutrition et le régime alimentaire de l'enfant. Parce que c'est capital d'expliquer aux mamans que l'allaitement maternel exclusif est important jusqu'à 4 à 5 mois en DIAS. Et également qu'il est également important d'éviter tout ce qui est sucrerie en général. Nous mettons également un accent sur tout ce qui est en lien avec la prévention des accidents et blessures pour l'enfant. Il est important de revoir le contact de l'enfant avec des animaux et de revoir également comment l'enfant joue dans la maison. C'est important. Nous parlons également aux mamans tout ce qui est en lien avec le lavage des dents, non seulement de la maman elle-même, mais aussi des membres du foyer. Parce qu'en fait, de façon pratique, on distribue des colgates et des brosses à dents dans les ménages où il y a des ménages qui sont en fait dans le groupe d'intervention afin que chaque personne de ce ménage puisse avoir une brosse à dents renouvelable chaque mois. Donc, ça, c'est un aspect qu'on insiste également là-dessus. On insiste également aux mamans sur tout ce qui est en lien avec les visites médicales. Quand est-ce qu'il faut consulter si l'enfant est malade, reconnaître les signes de danger, faire appel également aux médecins investigateurs pour éventuellement orienter ces bébés vers des structures qui peuvent les prendre en charge efficacement. Et on insiste également sur les mamans l'intérêt de respecter le protocole de visite qui figure dans le suivi. Pour ce jour, donc, nous avons au total 446 inclusions, dont 225 dans le groupe intervention et 221 dans le groupe contrôle. Nous avons 12 abandons. En effet, les abandons sont de causes variées. Et il y a, par exemple, la première cause d'abandon, c'est beaucoup plus des déménagements, en fait, de ces couples mère-enfant vers des villes hors de Bangui, ce qui fait qu'elles sont finalement inaccessibles. La seconde raison, en fait, des abandons, c'est aussi lié, comme nous le savons tous, un peu à un aspect culturel. Nous savons, comme je l'ai dit tantôt, il y a un prélèvement sanguin des bébés à 6 mois et à 12 mois. Certaines mamans n'acceptent pas du tout. Mais aussi, il y a des raisons qui sont personnelles et qu'on ne peut pas aller pousser plus loin pour comprendre de quoi il s'agit, parce que c'est en fait le droit de chaque maman de retirer son consentement qu'elle le souhaite. En conclusion, donc, force est de constater qu'en fait, AFRIBIOTA représente une expérience particulièrement enrichissante car elle associe à la fois recherche clinique, santé publique et innovation scientifique. Au-delà de l'objectif principal portant sur la croissance infantile, cette étude, en fait, a contribué au renforcement des capacités en République centrafricaine. Moi, particulièrement, j'ai bénéficié d'un financement pour une formation de master en santé publique. Et également, je suis candidat pour poursuivre en thèse, toujours avec le support de ce projet. Au-delà de ça également, ce projet permettra en fait à la République centrafricaine d'avoir une biobanque de grande valeur scientifique. Et au-delà de la formation que j'ai reçue, les relais communautaires aussi bénéficient en fait sur le terrain lors de nos descentes des formations, parce que tout ce que nous faisons, nous essayons de leur expliquer pour qu'ils puissent aussi se développer personnellement. Nous attendons donc les résultats finaux de cette étude afin de déterminer si cette intervention peut contribuer de manière significative à la prévention du retard de croissance chez les nourrissons en République centrafricaine. Nous vous remercions.
Oui, c'est Philippe Sansonetti. Je reprends la parole une seconde pour vous remercier tous les deux, Violetta et Dr Henko, pour vos présentations qui étaient extrêmement claires et rigoureuses. Merci infiniment.
Yes, so sorry, my microphone was gone. So if you have any questions, please feel free to ask us. I'm just trying to check if there are some questions on the chat, maybe.
Everyone, if you have any question, you can raise your hand and we will allow you to enable your mic. Or you can also click on the Q&A section and write your question. You can raise your hand and we will enable your mic. Or you can write your question in the Q&A section. Feel free. And while you are thinking about any question, we are reminding you that you have the PN link, community.pasteurnetwork.org, where you can follow every monthly seminar to know in advance the next topics. And every past seminar will be available on YouTube. Yes, Dr. Gopal, I allowed your mic. You can unmute yourself and ask your question.
Hello, I'm Deshmukh Gopal from the Institut Pasteur of Paris. I used to work on bacterial resistance in structure biology by doing the structure of recombinases, the enzymes that move recombination cassettes between bacteria. I had a question about the reversibility of dysbiosis. You said, Dr. Violetta, at some point that the Italian prisoners, when they went back, they had a spontaneous recovery. Is that something that can be used as a means of overcoming the disease? Is to find again some environmental conditions where you have the same ecosystem?
Well, first of all, I would thank you for your question. And without any doubt, the problem is that people that are living in countries where the hygiene measures are not implemented everywhere, it's really difficult to get that, because there is a SHINE trial. I don't know if you know it. It was a very important trial that insisted on hygiene measures and wash measures. Wash is water, sanitation, and hygiene. And it didn't show a real success in not only preventing stunting, but also in improving it. So honestly, that was kind of a failure for us all, because we didn't expect these results from the SHINE trial, meaning that on the field, wash, like water, sanitation, and hygiene measures were not sufficient. the question, if you ask yourself why, I would say, well, I don't have all the answers, but I have one. Because maybe it's too late in the sense that once the dysbiosis is established, at between 2 and 5 years, like the harm has already been done for regarding the epithelium, the gut epithelium, and it's easier somewhere to prevent an aberrant colonization than that to heal it. But I'm not sure. We only know that these hygiene measures between 2 and 5 years on the field haven't shown that it's really a bottleneck effect. They reduce it, of course, but not really significantly. And if you work on my antimicrobial resistance, something that we got also from our results in Mythica, and it was very interesting, was that, but that has already been described, that when there's a dysbiotic microbiota, like Klebsiella species harbor more IRGs than Bifidobacterium, for instance, and we have higher resistant burden also in the Central African Republic than infants who have more an eubiotic microbiota with enriched in Bifidobacteria, for instance.
Violetta, can I add a word?
Yeah, of course.
This is a very important question, and I would like to just put it maybe more in a perspective.
Yeah.
This disease that occurred to adult people was, called tropical sprue, and I must say that the time for recovery, where people were coming back to their metropolis in good hygiene condition, sometimes was several months or years. And many of these people remained with, sequelae like, intestinal discomfort and so on and so forth, even though their recovery was in general okay with regard to, weight and nutritional conditions. But the other point which is important is here we are talking about a different population. We are talking about very young kids. We're even talking about fetus in the body of mothers who are malnourished themselves. And this, is to be seen in a different perspective compared to adults, because these kids at these different ages have to go through checkpoints of development, whether it's intestinal development, whether it's neurodevelopment, and so on and so forth. And if they don't meet these checkpoints at the right time, I mean, they may have a hard time to recover. So I think the story is that this population is much more vulnerable, and so we cannot necessarily compare the two models, the adult and the child model.
Yeah.
Thank you. Maybe as a complementary question, then I would ask, just like you have the development of the tooth system in human at different lifetime points, do you think it's part of public education to teach how to be careful at different age, to be careful about some kind of nutrition habits? Infants were being taught in your program, but how about adolescents or young adults? Because at these different time points, people change their eating habits because of social interactions, because of schooling, changing of schools and university and so on. Is there, other than going to the dentist once a year, I think there's not many recommendations about oral hygiene.
Yeah, for pregnant women, actually, it has been already described in literature and in classic epidemiological studies that pregnant women should really make an effort and wash their teeth after each meal, because certain pathogens of oral sphaera are intimately associated to preterm birth. Like the way in which it's produced, we don't still know. We don't know if it's really absorbed and then microbial translocation, because you know that during pregnancy, the tight junctions of the gut are a little bit wider to be able to absorb more easier the nutrients. But also that means increased microbial translocation. So we don't know if it's via this way or hematic. We don't still know, but it has been already described that you can find the exact same isolates of certain bacteria from the mouth in the amniotic liquid of certain and the placenta, of certain preterm births. And of course, that's a pathogen thing, but it's also linked to, for instance, the presence of Fusobacterium. And Fusobacterium was exactly the same genera that we found here in AFRIBIOTA 1, associated with stunting later on. So during pregnancy, it's also a crucial moment in which oral hygiene should be encouraged. And of course, as you said, adolescents, because there's all this growth, as Philip said, this growth ways and time points. But I have the impression that oral hygiene in general in public health has been a little bit neglected compared to, I don't know, other areas such as gynecology or etc. And we shouldn't underestimate actually its effect. I don't know if you, Philip, you want to add something to that?
No, I perfectly agree. I would just add something. One thing is oral hygiene, and this is essential, and this should be part of education at any level. I mean, we cannot claim that AFRIBIOTA can take all this burden. I mean, what we would like is, and I think it's on the way actually, due to the good connection, especially in Bangui, between the Institut Pasteur and the health authorities, the Ministry of Health, and so on. So, I mean, this has to be extended under the responsibility of the educational system and the medical system. The other issue is, I would just like to stress it as well, is also the nasopharyngeal issue. Because when we saw this small intestinal bacterial overgrowth, of course, we have this predominance of microbial elements, pathogens from the mouth, but also from the nasal cavity, like Moraxellacea and other of these microorganisms. And these kids often have rhinopharyngitis, which are not so difficult to treat, but they at least necessitate what Violetta, what you said, which is nasal washing with sterile saline on a regular basis. Because if all these microorganisms, the one from the mouth, the one from the nasopharyngeal cavity, are permanently regurgitated by lack of hygiene, I mean, that's probably why they accumulate in the small intestine. I mean, they can survive gastric acidity and reside on the first area that is, I would say, friendly for their development. So all this has to be turned into, elements of education. So again, I totally agree with the question as it was asked.
Yeah, and the last thing also, of course, mucus. Kind of I saw a presentation about that also in one Pasteur thing, and the role of mucus in kind of protecting bacteria in the stomach. It was really interesting.
Very important.
And maybe I'll just switch to French for the Dr. Enco. Dr. Enco, vous êtes là? Parce que comme la connexion n'est pas très bonne. Super, on parlait, on disait justement et que c'est dommage que par exemple en Afrique centrale, il n'y ait pas de programme de santé publique sur l'hygiène orale en fait. Et il me semble qu'il n'y a aucun pays de l'Afrique centrale où il y a un problème, où il y a un programme genre de l'État en santé publique orale, c'est le cas?
Non, je n'ai aucune information par rapport à ça, du moins en Centrafrique, non. Vous m'écoutez?
Oui, on vous a entendu.
OK.
Docteur Enko, je ne sais pas si vous m'avez entendu tout à l'heure, mais je voulais vous remercier très sincèrement pour votre présentation qui était complète, rigoureuse et avec toutes les perspectives, y compris de formation locale. Donc, merci beaucoup.
Merci beaucoup pour ça.
Thank you, Dr. Enko, but also thank you, Dr. Sansonetti, Dr. Violeta Moya-Alvarez. Thank you for your time and for sharing with us all about this wonderful program. So I want to remind you that our next session will be on July 9th. The monthly seminar is on every second Thursday of the month. So you can join PN Link today to be the first to know about our next topic. It will be announced before being launched on our social media. It will be launched on PN Link. So join PN Link today to be among the first. And if you want to replay this session or every session that was passed, you can be on the Pasteur Network YouTube channel. So see you on July 9th, and thank you and have a good day. Bye.
Thank you.